Hello, and welcome to the show. Today, we're diving into the fascinating world of archaeogenetics, a field that uses ancient DNA, or aDNA, to rewrite human history. Imagine being able to read the genetic stories buried with our ancestors, tracing their journeys and interactions across thousands of years. That's precisely what archaeogenetics does, blending genetics with archaeology and anthropology. So how does it work? Scientists carefully extract tiny amounts of DNA from ancient remains. Think bones, teeth, even preserved tissues. This DNA is often very old and broken down, making extraction incredibly difficult. Contamination with modern DNA is also a huge hurdle, especially when working with samples from warmer climates where DNA degrades faster. But thanks to advanced lab techniques like PCR, and especially next-generation sequencing, these fragile fragments can be amplified and read. Powerful computer tools then help piece together the genetic puzzle, comparing ancient genomes to each other and to modern populations. What are we learning? One of the biggest impacts is on understanding population history and migration. European prehistory, for example, has been completely reshaped. We now know about distinct waves of people, the original Mesolithic hunter-gatherers, the early farmers who migrated from Anatolia, and later Bronze Age groups arriving from the eastern steppes. These studies show how modern Europeans are a mix of these ancient ancestries. But it's not the same everywhere. Studies in southern Africa reveal incredible genetic continuity over 9,000 years in some areas, a stark contrast to the population turnover seen in Europe. Research in East Asia is also revealing complex histories and migrations that formed modern populations. Archaeogenetics is also brilliant at detecting admixture when distinct groups met and interbred. This gives us direct proof of interactions sometimes invisible in the archaeological record. Perhaps the most famous examples involve our archaic relatives. We know that modern non-Africans carry DNA from Neanderthals and Denisovans, evidence of interbreeding as early Homo sapiens expanded out of Africa. The main Neanderthal mixing event is dated to around 45,000 to 49,000 years ago. We're also learning about more complex interactions with different archaic groups, especially in Southeast Asia and Oceania. Within more recent human history, we can track the mixing of migrating farmers and local hunter-gatherers during the Neolithic transition, and follow later movements, like those associated with the Bell-Beaker culture in Bronze Age Europe, or the Anglo-Saxon migrations to Britain. The applications don't stop there. Archaeogenetics helps trace the domestication of plants and animals, identifying their wild ancestors and tracking the spread of agriculture. It even allows us to reconstruct the genomes of ancient diseases, like the plague or tuberculosis. This helps us understand how diseases evolved and impacted past populations, possibly even shaping our own immune systems through ancient pandemics. The field is growing exponentially, with more ancient genomes being sequenced worldwide. While Europe was an early focus, research is now global, moving towards incredibly detailed local studies. We're gaining insights into past social structures and even family relationships. Archaeogenetics truly offers a powerful new lens on the intricate story of human migration, evolution, and history. It's constantly refining, and sometimes completely overturning, what we thought we knew about our past. Thanks for tuning in.